Un, dos, un, dos, un, dos, un, dos.
Yeah. Is it off right now? Can I put it off?
ดีครับ approach is environmental DNA, that's very important. We have a very, very, very huge expertise. Very interesting. So,
Oh, there we go. Perfect. Thank you.
just before, if there are some questions in the talk from Nicole, just ask the hand, nothing more. If anyone have problem with the English, maybe you can make a question in Spanish and I'll make the translation. Okay.
Okay, perfect. Yes, thank you for the introduction. And I'm going to be talking about how we're using environmental DNA to support both conservation and management of marine species. For those who were in the workshop yesterday, there'll be a little bit of repetition of some of the basic information, but that's not a bad thing. Maybe it'll sink in a bit more. But I'm not going to get too much into the details of the methods here since we did that yesterday. But here I really want to show you the diversity of types of research we can do using this technique. So if you weren't here yesterday, environmental DNA, this is DNA that's released from organisms into their environment. For the work that I do, I sample aquatic environments, so I'm taking water samples. So in this case, we're talking about organisms that might be shedding mucus or scales or blood or feces into the water. And then we collect that environmental DNA in our water samples. Now, there's two broad approaches to this research. The first is called metabarcoding. This is when you want to know about all the different species in your water sample. So here you might use universal fish primers and identify or try to identify every species of fish found in that area. The other type is targeted species detections. This is where you develop a genetic assay specifically to target DNA from only one species. And in my lab, because we predominantly work on threatened rare species, we use targeted species detections. So the general process is you go out and you collect some water samples, just like this, and then you filter them. So for the filtering, this is your water sample. This is a pod, and there's a piece of filter paper in there. This is a vacuum pump. You turn this on, and it pulls the water sample up. It filters it, and all of the sediment and eDNA gets bound to this filter. Then you take the filter. So this is a picture of a filter with a lot of sediment from the water. There'd be a lot of eDNA here. And we cut it in half, and we conduct a DNA extraction on half of that filter. Very similar to how you would extract DNA from tissue. Then we screen that sample for the target DNA from whatever it is we're looking at, and we use PCR for this. We specifically use what's called droplet digital PCR or ddPCR. Now, the reason why we use this technology is because it is very, very sensitive. It can detect just one copy of DNA in a reaction. You don't get that level of sensitivity with other types of PCR. How this works is it will take your PCR reaction and you add an oil to it, and it creates up to 20,000 nanodroplets. And then the PCR occurs in each droplet. So you could think about it in terms of in one reaction, you have up to 20,000 PCR replicates. If the droplet does not contain the target DNA, it does not fluoresce, and it shows up in the graph as a gray dot here. So this band right here that's very, very thick, those are actually individual dots. It's just that there's so many droplets that do not contain target DNA. If, however, the target DNA is present, it has an internal fluorescence probe, and that creates a fluorescent to appear, and the machine will then read that. And so each of the droplets that contains target DNA shows up as a blue droplet up here. So this allows, even if there's just one single copy of your target DNA in one of these droplets, we can detect that. And so this here just shows some dilution factors of some DNA samples that we ran. And you can see that we get down to just one copy of DNA. So one of these droplets that contains DNA. And that corresponds to 0.25 picograms of DNA. So this is incredibly sensitive technology. There's a lot of benefits to using eDNA. So as someone who uses genetics to study marine species, usually the other methods include things like going into the field, putting nets in the water to try to collect species, maybe tagging species to get information. And so in comparison to those approaches, which we call traditional methods, this is much more sensitive. If you're looking for something that's very rare or uncommon, your best bet is to collect a water sample and screen it rather than putting a net in the water. If you could do net surveys for months or even years and get nothing, we could go out there once and detect it. Because it's so sensitive, this also makes it cost effective. Again, when species abundance is low. So if you have very few of a species and it would require a lot of survey effort to find it in the wild, then you should be using eDNA surveys. It's also non-invasive. I work on threatened species, and if you want to collect them in a net, that can injure or cause stress to the animal. And it requires no permits, which is one of my favorite parts. So it sounds almost too good to be true, this amazing technology. Why would you do anything else except eDNA surveys? Well, it's very challenging work. So there's not a lot of eDNA from any single animal in the environment. Think about how diluted that would get in the marine system. One animal releases DNA and that quickly gets diluted. So it's low copy. And because of sunlight, the fact that it's not in preservative and microbial activity, that DNA degrades. So it gets smaller and smaller and smaller. And it's moving in the water column. So that makes it really unlikely that you're going to capture what you're interested in. The second major challenge is the risk of contamination. So this might be from target species. So if you're doing eDNA work, you should not have your target species in the boat or in the lab. We're also worried about carrying DNA with us between sites. So if at site 1, the target species is present and some of that water through waves splashes on the boat, and then we go to site 2, there's concerns that we've carried DNA and then we might get false positive detections at site 2. So we have extremely rigorous protocols in order to prevent contamination. We clean everything with bleach, and we have a lot of negative controls. We have a lot of lab spaces where only one activity happens in each lab space. And we have what's called unidirectional workflow. So what that means is samples enter the lab, DNA is extracted here, and then it moves forward to PCR, and then it moves to analysis. It doesn't move backwards in that workflow. So we are using this concept of going out and collecting water samples and looking for things of interest to investigate a lot of different questions. So it's not just, is something there? Yes, we're looking for presence of species. We're also using it to monitor species recovery. We're looking at seasonal migration patterns in marine species, identifying priority locations. We're looking at spatial patterns of habitat use. So if you've ever watched Shark Week or something, you know, they usually put tags on an animal to see where they're going. Well, we're actually using eDNA to see where they're going. Changes in distribution related to climate change, abundance of species, and we can even look at historic trends in abundance, and that's through the use of sediment cores and water samples. So these are the types of questions that we're answering in my lab, and that's, I think, really important to understand. We do study eDNA as a technique and as a method, but mostly we use it to achieve or to address questions in conservation. So I'm going to just highlight a couple of our projects that sort of show the versatility of this tool. And it also shows all the great places I get to travel to sometimes. So in my lab, we have done eDNA projects on almost 20 species in the last 3 or 4 years. So we've been extremely busy, and you'll notice it's not focused on just one taxonomic group. I do have preference to sharks and rays, which are these two, but that's not all. We have work on marine bony fish, freshwater bony fish, turtles, terrestrial and marine mammals, and even amphibians. So we work in almost all vertebrate groups. Today, I'm going to share some of our projects that include rays, marine bony fish, and manatees. And the first species, I've been talking about these sawfish now for 3 days because we have a lot of research on sawfish because they're one of the most threatened species on the planet. So we are trying to get as much information as possible. So this is the small tooth sawfish. It has this huge saw on the front of its face. It is an electroreception organ. So it detects the heartbeats of fish, and then it can slash at fish for feeding. It's critically endangered. What that means, it's basically on the verge of extinction. If we lose it, or the next step, if things get worse, is going to be extinct in the wild. So again, this is a very severe situation. This is the historic range of this species. So it was found in the Atlantic Ocean, but it has experienced significant declines. And this is the only place where viable populations remain. And it's really hard to see, so we have to zoom in here. It's predominantly in the US. There is a small population in the Bahamas, but it's thought to be much smaller than the population we have in the US. And the US protected it in light of this, and in light of the significance of the population there in 2003. Now you might be wondering, why on earth did they decline to this extent? 2 main reasons. One is habitat loss and degradation. Sawfish, and especially juvenile sawfish, live in shallow coastal waters. They spend their early years in nursery areas in the mangroves. Where do people like to live? Where do, we have ports, we have industry on the coast as well. That's also where a lot of fisheries are. So we have extensive habitat degradation. This is actually in Florida, And you can see there's this tiny remnant of mangrove habitat left. And all of the other mangrove has been cut down, destroyed, and instead we have communities and canals being built. So this has been a big problem. The habitat doesn't exist anymore for small-tooth sawfish in many areas. The other reason they've declined is fisheries. We have there were direct fisheries to target sawfish in some countries for the fins and for the meat. But by and large, and definitely in the United States, the biggest cause of mortality was bycatch in fisheries. That is when they're accidentally caught because they're easily entangled, and then the fisher would often take them. Now, within the US, here's our map, and this is what we call it's called the core range. This is that area where you can still find sawfish, and sometimes fairly easily. Right on the side of the road, you can walk out and there's some baby sawfish there. But while they were historically found from the Carolinas up here, all the way around the Gulf of Mexico to Texas to the Mexico border in the United States, their range declined to only this green part of Florida by the 1980s. They were basically locally extinct everywhere else. Now, I said they were listed in 2003. That was 20 years ago. And that's been 20 years of research, public education, and legal protection. And this is kind of it's a moving in a success story direction, because now in Southwest, populations are no longer declining and they're actually doing better. In addition, we have this what's called public encounter reports. So we teach people about sawfish, and if they see them, they call a hotline. So we have a 1-800 sawfish phone number, and fishers or general public will call in when they see them. And all of these red dots are these reports. And you can see there's a lot of reports in areas where we thought this animal was extinct. But you can't necessarily trust these reports, because sometimes people think they saw a sawfish and they actually didn't, or they're just telling stories. We call them fisherman stories. So we need to have some scientific evidence that these animals exist in these locations. You could put nets in the water, but that's not very cost effective. So we decided to do some eDNA work on this species. And we have surveys right here. This is Tampa Bay. We decided to go here because it's very close to where sawfish populations are doing quite well. We also went on the other side. This is called Indian River Lagoon, but I live up here. And so these were really interesting to me. And so we also did some surveys here, very far away from where we know we can find them. The first step, of course, was to develop an assay. So this is what we talked about yesterday, how this is done. So we developed a genetic test that would only detect sawfish DNA and nothing else. Once we thought we had something that worked, we had to test it. So we went to Florida and we collected water samples from an estuary where we knew sawfish were present. But at the time, we didn't know if that would actually work. So we caught a baby sawfish and we put it in a tank on the boat, and then we collected water samples from this tank. And so these are the data. This is just a negative control. This is what the water sample looked like that we collected. So we've got lots of target DNA. And from the tub we had, at about 0 minutes, we had an incredible amount of sawfish DNA. And at 30 minutes, it was almost too much and overloaded the machine. So once we had this showed the technique worked. It showed, yes, we can take a water sample and detect DNA from it. So these are our data for those areas that we surveyed. So we only consider a sample to be positive for DNA. So we can be very, very confident that an animal was there if it meets 3 analysis criteria, which here is in a red diamond. And you can see there are no red diamonds on this map. So this was a little bit, it was disappointing to see this. And this is over 2 different years of sampling. But it was corroborated by field surveys. So I also had a collaborator at the time out there putting nets in the water who also caught nothing. Now we sampled in August, which is our summer. And later on, a few years later, maybe I think in 2022, a paper came out from tagging data that said they may not be here in the summer, they might be here in the winter. So we're planning on going back in the winter to see if we can detect them. On the east coast of the US, so this is over here, we had some more positive data. So again, this is summer over 2 years, and we had 4 positive detections in 2018, and we had 1 in 2019. But there was some skeptics who were like, I don't know, is this real? I don't know if I believe you that there's sawfish there, but it was great because then later in 2019, a collaborator caught one and put a tag on it. So that helped, I guess, reinforce that these data are good. And then this is closer to where my lab is in Mississippi and very, very far from this area where we know sawfish exist. So it was believed the sawfish were gone from Mississippi waters since the 1980s. And we went, here we had we went every single month because it was nearby, but we weren't able to go out in 2019 because of poor water quality conditions. But in 2018, we didn't get anything in June, nothing in July. Finally, in August, we had 3 detections. And again, there was some skepticism, but the following year, a sawfish was caught and photographed in this area. So again, helping to support that these data are real. So in Mississippi in particular, this had significant conservation outcomes because we didn't think they were here anymore. They weren't protected in our state. So you could still sell, technically, sawfish parts within our state. So we're working to close that loophole. And so we're working right now. We have a draft list done, and it will be protected in the state of Mississippi now. And we have all of these signs put up to help educate people. So this was one of our early projects on eDNA. Now what we're doing is, okay, sawfish are up here in the summer. Are they there in the winter? We have no idea. It gets surprisingly, it gets quite cold here in the winter. The water gets very cold and sawfish like warm water. So we don't know if they stay or if they leave, but we need to know this. So we've got seasonal samples that we're currently analyzing in order to address seasonal occurrence and migration in the species. And another thing that we're working on with this project is, just because you collect a sample that's positive in one location doesn't mean the animal was right there. Because remember, marine, large systems with currents, tides, and waves, as soon as that DNA is released, it's moving around. And so we're using modeling approaches to estimate where it may have come from, depending on the rate of degradation. So this is just from a paper that shares that same idea. Here they got a detection at the station M1, and when they apply a model that uses a fast rate of eDNA degradation or a moderate rate, you can see it really doesn't make a big difference. They're able to say it came from somewhere south of this point. So we're looking at doing something similar. I do not think that eDNA went from Florida to Mississippi. I think it originated somewhere in Mississippi, but we need to know where so we can protect the habitat. We're also, so that was, you know, it's there. We're using it to monitor recovery and looking at seasonal migration. We're also using eDNA to identify where conservation efforts should be focused. So in conservation, there is very little funding for the amount we need to do. And so we have to prioritize and say, you know, where are we going to put the dollars for any particular species? So for this one, I want to highlight large-toothed sawfish, which is a species I've been working on for almost 20 years now. This species used to be found all around the world in the tropics and temperate latitudes. But again, because of mortalities in fisheries and habitat degradation, it's experienced at least a 61% reduction in range. The only place you can find these really reliably in good numbers is Northern Australia. Now, I have a PhD student in my lab who's working on population genetics of this species using DNA that's captured from these old saws. And what her work has shown is that the large-toothed sawfish in this Indo-West Pacific region here on the map are genetically a distinct lineage from those in the Atlantic and the East Pacific, which is over here. So they're very, very different. They used to actually be considered separate species, but then they lumped them. But here we know that this genetic lineage is secure because we have this population in Australia that's doing quite well. What's really concerning, especially given the taxonomic, they're different, they're the same arguments over the years, is this area. And the risk of what is going to be lost from this species if we do not identify and secure a population in this part of the world. So because we don't know, so a lot of these these blue areas might be one report in 30 years. And that doesn't mean that you should dump a whole bunch of money in one area with one animal in 30 years. So what we're doing is we are going to these areas and doing eDNA surveys to see what we come up with. And we started in Panama and Colombia. So this is a colleague of mine. His name is Juan. And I met him in Brazil in 2018 at a conference, and he said, I'm really interested in eDNA, but I don't know how to do it. And I said, well, we know how to do it. Let's work together. And so he got a grant to go to the Darien region of Panama and also Colombia to get some samples. Then we had the opportunity through a funder to go to the Gulf of Chiriquí, which is here in Western Panama. And so I called him up and I said, why don't you come? I'll train you, and then you can go do your surveys. So the main reason for selecting these countries first, because we thought it was going to be the most likely. There's a lot of publications that say the habitat's there, they're protected. This is the place to go. So we went. This was our field team here. We had 5 scientists and a boat captain, and this served to train Juan in these methods. This is where this is filtering. It was absolutely stunning country. This just shows the habitat. Can you play the video? So this is exactly what a baby sawfish wants. These beautiful, pristine mangrove habitats. And it just stretched on for miles or kilometers and kilometers and kilometers, these labyrinths of mangrove. So we were really excited and we thought, yes, we're going to get positives. We were taking bets on how many positives we were going to get because the habitat was there. Unfortunately, there's also a lot of fishing. these are our data. We had 100 samples and we did not get one positive across any of these areas. And this, you can see how densely we sampled this area on the Gulf of Chiriquí. And here it is zoomed in. We had really good coverage in this area as well. And then we had nothing here and nothing here. This was extremely disappointing. But this is what happens when you work on threatened species. It's not always good news, but we're not giving up. We are now working with collaborators at the Smithsonian Tropical Research Institute in Panama City, Panama. And they have collected more than 600 samples in 2022 and 2023. And so these are the areas that they have samples for. And so in 2024, a student from Panama will visit my lab and analyze the data for the samples that she has collected, and fingers crossed. I don't actually have a slide for it, but I also have a collaborator in Peru, and they will be collecting some samples in January of next year to also be screened in my lab for this species. And we also have surveys in Mauritania and Guinea-Bissau in West Africa, again, through collaborators who are going out, collecting these samples, and then we are screening them. So, and then I have a student going to Brazil next year as well to do some training over there. So we have this huge network of different collaborators and researchers all working together to try to find perhaps a population of this species. We'll see how it turns out. Okay, let's look at how we use this for broad spatial patterns. So for these, I actually have results. These are all sort of in progress at the moment, but I just want to share them because some of them are really interesting and unique projects. So the first one is looking at broad spatial patterns of habitat use. So what does this mean? This means, is this animal using coral reefs or seagrass beds or mangroves or deep sea? Because you might think this is basic. Don't we know this? No, we don't know this. For many, many species, we don't know where they even are in the water. And this is the clown wedgefish. It is a species of ray. It is also critically endangered. This one has an interesting story. So records for this species were only from the 1930s, the 1970s. There was one record in 1996, and the species wasn't even described as a species until about, I think, 2016. Very, very recent. And in 2019, 3 years after we said this species exists, the paper was being written declaring it extinct. So no, to date, no shark or ray has ever been declared extinct. It's coming very soon, but at this point, that had never happened. Also in 2019, though, one showed up as its extinction obituary was being written. One showed up at a fish market in Singapore. That was great. Okay, it's not extinct. But a fish showing up at a fish market in Singapore tells you nothing about where it's from, because these fish are coming in from throughout Southeast Asia into this fish market to then be sold. So this was great, kind of. We didn't know where it came from, but it wasn't extinct. So that was still a plus. Then in 2020, Matthew McDavitt, who actually has no formal scientific training, he is a lawyer, but he has a passion for conservation, and he is a self-trained scientist, and he was scouring Facebook, Instagram for records of species. He always does this because he documents trade. And he found one in 2020 on Facebook, I think it was, which was from these islands right here. So here's the islands. They're just off of Sumatra, these two little islands. We still didn't know where they were from, though. We knew they were from these islands, and we know that these islands are not using large fishing boats. So we could assume it was from somewhere nearby. So given this, species went from extinct to one showed up to a couple more showed up, and we're narrowing down where it might exist. We wrote a proposal which got funded to go and do eDNA surveys. We went to Singkep Island, which is right here. This one right here. Extremely remote. It took 5 days for me to get there of traveling. So we conducted these surveys in June 2023, and we met up with some local researchers from a university in Indonesia, and I took them in the field for, I think it was 3 weeks, and trained them on how to do these surveys. We did very well. We sampled, this is coral reef. We sampled coral reef habitat. We sampled rocky shores. We sampled islands, and we sampled, this was remnant mangrove. This is the only mangrove that's left on this island, is this sort of patchy mangrove habitat. So we don't have any data yet, but this is just a map that shows we got complete coverage. We collected 100 samples, complete coverage around the island to try to pinpoint where this thing is and what habitats it is associated with. This was the most challenging field work that we have ever done. It was so incredibly remote, but we accomplished it. So we'll see. We just did one island though, so we may in the future go back and sample this other island, Lingga, but that will be even more complex because there's no roads. So you have to go all by boat. And we may go to Eastern Sumatra as well and see. Depends on our data. Okay, just 2 more examples. This one is changes in distribution associated with climate change. And this one is incredibly pertinent today, because when I say climate change, what do I mean? Our oceans are getting warmer, and they're also becoming more acidic. And this is having a lot of impacts on ecosystems and on species. Species might find themselves suddenly in waters that are too warm for them. And temperature regulates basically all of life, right? Through enzymes, function, metabolism, everything is linked to temperature. On the flip side, some species may be able to move into different waters. And so there's a lot of research on the impacts of climate change on marine species and ecosystems. It's all very interesting. That could be a whole nother seminar. But we are specifically looking at climate driven. So that just means that temperature changes in distributions of the manatee. So manatees are changing their distribution because the waters are getting warmer. And this is actually a case that is perhaps a positive thing instead of a negative thing, which is what we usually see with climate change. So this is their distribution. So this is from I borrowed this from a paper, just a nice, very rough graphic of this is where the West Indian manatee occurs. And you can see it occurs in Florida. It loves warm water. I mean, it gets hot in here in the summer, but winters are cold. In 2009, the first manatee, which was named Bama, was actually caught and tagged in Alabama in the northern Gulf of Mexico. So this is right here, and our lab's up here. So this was in 2009. This was huge. This made all the news. Why is there a manatee somewhere we don't expect to see a manatee? And it's because of what we call tropicalization of the northern Gulf of of Mexico. The northern Gulf of Mexico is becoming more and more tropical. It's getting warmer and warmer. Our summers are becoming almost intolerable, and the water is very, very warm. And so because of this, it's thought the West Indian manatee might be coming and might continue to use these waters more and more. And it could be a regular thing. So right now in Florida, you want to go see a manatee, you just go down to a dock and you see a manatee. That could someday be exactly what happens in the northern Gulf of Mexico. So what does, and this, I put this photo in here. This is what the water looks like. It is not Florida. This is a tag on a manatee. There is a manatee about this far underwater, but you can't see it, right? You cannot see it if it were not for that tag. For that reason, we are looking at using eDNA to figure out when they're coming, how long they're staying, and what areas they're using. And then we want to correlate manatee occurrence to our eDNA data. So we've gone out and collected samples from all of these locations. We sampled in 2022 and 2023. We use an assay that was already published. So here's a beautiful ddPCR assay. We had to validate it a little differently than we normally do. We didn't put a manatee in a tank for this one. I actually got a jar. This is a little disgusting, but we got a jar of manatee poo. We put it in a tank and we shook it up, and then we took water samples. So at my university, I get packages that say manatee poop for Dr. Phillips. So anyone who thinks marine biology is glamorous, I just show them that picture and I'm like, no, it is not. So we have the 2022 data in, and we got all these detections. So the green here are positive eDNA detections of manatees. And we have these 3 stars. These are validated positives. So at these sites, we actually saw manatees. No manatees were seen at any of the other sites, but you can see we've got detections way upriver here and using this river as well. So this was great. This was in September. So this is getting late summer, early fall for us. So this is really a pilot study to then say, this works. We need to regularly, maybe monthly, go out and collect samples to monitor manatees' habitat use in these waters. And even since we started this project, now manatees are showing up in my state of Mississippi, which is one further west. Okay, and lastly, species abundances. This is species abundances with eDNA is some pretty intense work. So this question has been asked for years and years and years. We know we can get presence absence, but what about when we quantify the amount of DNA? Can we use it to estimate abundance and say there's more or less of something? In the freshwater eDNA literature, the answer appears to be yes. So this is graphics from freshwater studies where you have biomass or density of animals, and you've got the eDNA concentration. So this on the y-axis, and basically as the density or the biomass increases, you have greater quantities of eDNA. But this is freshwater. This is a tank. This is a lake. This is a pond. This is not the marine system. And so we're working on this project called the Great Tur Amberjack Count. It's a very large project in the US funded by the government. We have 13 institutions involved and 18 PIs. And each of these PIs has different expertise or works in a different area. The goal of this project is to estimate the abundance of the greater amberjack, Seriola dumerilii in the US, Atlantic and Gulf of Mexico waters. Why? Fisheries. So this is where I switch to management from conservation, but it's still kind of conservation because it appears that this species is not doing very well. It's very important. It's very important recreational species. And there's commercial fisheries as well. They actually had to close the fishery early this year because there was so much concern over the stock, over the population's health. And actually, as I was flying here in the airport, in Texas, I overheard a conversation. Yeah, they closed that amberjack fishery. Rah, I caught an amberjack and I got in trouble and I got fined. And I'm sitting there like, yeah, yes, we're working on that project. This is what the species looks like in a photo. Now, okay, how many? I mean, that is an impossible question to answer, it seems like. How many fish are in the ocean? How many fish are there here? Well, this team has done something similar for a species, another species in the Gulf of Mexico. And they're using lots of different types of data. Population structure with SNPs and close kin analysis, cameras, putting cameras down and looking at fish, tagging them, just every kind of methodology you can imagine. But there's an eDNA component because the government specifically asked for this. And so they asked me to join their team. And we have specific goals. The first goal is to develop a multiplex assay to detect greater amberjack. This is what we're interested in. But they also wanted us to be able to detect 3 other species: Amalcojack, lesser amberjack, and banded rudderfish. These are the only Seriola species we have in the area that we're working in. And you might be like, why did you have to detect all of them? Why couldn't you just detect the one that you're interested in? Because a large component of the budget is going to camera data. So they're putting cameras down with bait, without bait, different ROVs, all kinds of cameras to try and get a picture of this. And this is actually one of the better quality still frames because they're trying to get counts from camera data, and sometimes they can't identify the species they're looking at. So if they know, if we're telling them from the eDNA data what our confidence level is that the lesser amberjack isn't there, for example, then they can help differentiate and assign species IDs. So this was our first goal, and we did this with ddPCR again. This was the first time anyone had done 4 species on ddPCR. I hired a postdoc to do this work. She knocked this out, as postdocs do, in about 4 months, which was incredible. So what you're looking at here, this is a negative droplet band again, just like the other data. But then instead of just one droplet population for one species, you have 4. And each corresponds to a different species. So when we screen our data, we can see how many positive droplets we have within each of these populations. And we can say, you didn't have any banded rudderfish, or you didn't have any Amalcojack. And we can also look at potentially ratios to say, yeah, you had a ton of greater amberjack eDNA and just a little bit of amberjack. And then they use that information. So this part is done for us. The other parts are more difficult. We have to compare eDNA quantification estimates of greater amberjack to their data sets. So maybe we have 5 copies per microliter. We're comparing that to how many they get through other means to see if there's any relationship at all. The other component we're supposed to work on is we're putting in those cameras and traps with bait and without bait. And the question is, what is the impact of bait? I mean, if you use bait, you want to attract fish, right? And so there's some concern about biases associated with data from baited cameras. So we collected eDNA samples before a baited trap went in and after to see if there was a change in the amount of eDNA, which then can be interpreted. They also want us to use negative data to support what are called sampling zeros. This is, they put a camera down for an hour and saw nothing. Is that real? Does that mean the fish isn't there? Or does that mean you just didn't see it? So we're going to use eDNA data to either say, no, there were fish there, you just didn't see it, or we didn't get any either. So we are, for those 3 goals, we have to do a lot of field work. This is my postdoc, Joanna. She came from Portugal to do this. She spends a lot of time on the boat offshore collecting water samples. And we have a little bit of data analyzed. So she's just finished screening her samples last week. And so we don't have the data in for everything, but we do have the data for these traps. We have data for before the bait traps go down and after to see if we are potentially attracting fish. So here before baited traps, we have the stars just mean there was detection. It's just very, very low. We have very low detections here. And after the baited traps go in the water, we're getting spikes in our quantities. And so we need to take this into consideration when we're comparing data sets from this type of data from baited traps and data from non-baited. We have to be very careful. But it's also causing me to question this whole thing about eDNA in abundance. Because what does it mean that there's an increase in eDNA? I mean, your brain automatically says, well, it means there's more fish, more DNA, more fish. But maybe it's also that the bait, what happens when you put bait in the water, the fish come in, they get it, their activity increases. They're swimming faster, they're bumping up against each other. We could just be detecting an increase in the eDNA shed rate and how much is being lost into the environment. So you have to be really careful. And I'm seeing a lot of papers come out that just says more eDNA, more fish. And I'm very skeptical after looking at these data because the camera data for this does not show an increase in the number of fish. So we'll see. So we're still analyzing all of these data, but we're working on the first paper from this with the assay and some basic experiments. So in summary, eDNA is extremely powerful to study marine species with lots of applications. It's not just I did eDNA on this species and that's it. There's lots of ways to use it. You have to have a lot of lab work, a lot of controls to make sure your results are valid and meaningful. And we need, and we're working on future research to resolve uncertainties about fate and transport. So that means what happens to it after it's shed, where is it moving? And this relationship with abundance, because especially fisheries and governments are interested in whether or not this tool can be used like this or not. And of course, the goal is to support better conservation. So I want to say thank you to my lab for working so hard on all of these projects. My collaborators, you have to work with a lot of different people when you work on threatened species. And if you have any questions, I will take them.
Thank you.
This method can apply in world ocean not close to Earth zones.
So you mean like offshore, far out into the ocean?
Yes.
Yes. Yes, absolutely. So the amberjack work that we are doing is very far offshore. Those depths are 50 meters, 150 meters. There are even people doing eDNA work at deep sea thermal vents, which are 2,000 meters. That's very, very difficult. But you can use this anywhere in the ocean if you can get a water sample. But it gets very difficult the deeper you go in the ocean to get those water samples.
Thank you.
You're welcome.
So just a final remark. Un poco lo que hace Nicole, un poco las que tenemos que tratar de traer gente fundamentalmente de química, sistemas moleculares, para poder acercarlos a temas de ambiental. O sea, hay un campo enorme para los químicos en esto, para poder trabajar en este aspecto. Es un tema que viene, lo único que va a hacer es crecer, que es un impacto ambiental tremendo, no solamente a nivel de especies como grados, sino en vertebrados y en algas. Eso es importante, un poco esa es la idea. Thank you very much, Nicole. We learned a lot of this day of your research. So we have a final closing.